I'm going to tell you a little bit of a story. So Mosaic is a drug discovery organization. And I'll tell you a little bit of a story in the beginning on how we came into existence and why, what we thought the need was. And it's basically around the idea that outsourcing has been going on for a long time in the drug discovery and development business. Even as early as 1940s and 1950s, a lot of preclinical research was getting done, animal studies and the like. And now it's advanced to, even in very large pharmaceutical companies, much of the manufacturing and clinical development, the vast majority of it is actually done by outsourcing, even in very large companies, certainly in small companies. But interestingly, the research side has really lagged behind in this regard. And it's just not that there's no outsourcing of research, certainly there is, but it's just not to the same degree. And recently, as a result of COVID in part, and some technology improvements in communication like Zoom, there was a bigger need during COVID. A lot of companies, especially on the East Coast and maybe on the far West Coast, where space is at a premium and you couldn't get as many people in there, you still needed to get the work done. And so people started looking for solutions. And this allowed us in Boulder, Colorado, where we have a little more space, to be able to keep going while some other people were struggling. And so we started recognizing a need that was solved by, in part, being able to communicate by Zoom and the like. And so the big question is, what has kept research, drug discovery research, as opposed to development, really from catching on faster and filling that need that sort of has started to evolve now? And it's because it's actually kind of complicated. Research is actually research. And there isn't a simple, relatively simple, straightforward engineering problem to be solved here, but it's multi-component, complex interactions that are going on. And that's what caused us to realize, well, if you could build an organization that could do this for a contract, that had end-to-end research capabilities and could interact on these multiple levels that was desired, and people focused on making medicines, not just making molecules, maybe there would be a way to solve this problem. And so what was started off as basically what people thought you had to do is you have to build a research organization. It's just too complicated to outsource. You're going to be spending all your time managing all these organizations, flipping balls back and forth. And so we're just going to build it ourselves. Well, that's a time-consuming process to build it at all. It could be a year before you're even up and running. And in addition, by the time you're up and running and actually get your results, then all the money of your company starts going into development. What are you going to do with all these people in research? I think we know what ends up happening. And so why do that? So that's in part why we created Mosaic. We have all the same people that you would hire to build your organization, but you can come work with us. We allow you to basically skip the whole step up top of building the organization, and just as soon as we have an agreement in place, we can start working for you and in a collaborative way with scientific interactions that you need. And then you don't have to build it yourself and wait for it to be done. So that's what we have. We have on-demand resources. We're there when you need us, and we're not when you don't. We see a lot more things than most organizations see, right? Most organizations are built to do one or a small number of things. Many, many different types of companies and biologies and therapeutic areas come through us. And so we see an enormous amount of work. And so there's a diversity of experience that we get that ends up getting applied to any new program that comes in. So you get these accelerated timelines, you get a much higher probability of success because we've seen so many more things than a traditional biotech company might have seen. And so in the end, we think we're more, well, that one kind of came out kind of funky, didn't it? we think you're a lot more likely to succeed with us because we're going to be working closely with you as scientific intellectual discussions, but the work is still being done by the very same smart scientists that you would have hired yourself. Okay, so who do we work with? Startup companies, early stage, sort of Series A, biotech companies, venture firms, or people just trying to see if there's a there and a first set of sort of seed projects, biopharmaceutical companies that have more work than they can do and so they want to outsource part of it, companies that have moved into development and had to eliminate their research for financial reasons, but now want to build a pipeline again, now come back to us rather than build it on their own. And of course, academic groups. Okay, so that's why Mosaic came into existence. That's what we do, a broad set of work. So I thought I'd just give you a few examples of the kinds of things that we do and the programs that we run. The first one I'm going to talk about is and because we're at Peptalk, we can discuss things that aren't just antibodies, even though I'm in an antibody section. I'm going to go out on a limb here. I'm going to talk about actually the first program we ever did before we were full-time in this business. We were working on an enzyme, a protease that specifically cleaved complement factor C3. And we were excited about the fact that there was this company called Apellis, you may have heard of, that had a C3 blocker. They use it systemically, but also for an intravitreal injection in the eye, sort of in the back of the eye treatment. And they were using it for geographic atrophy. And so we saw an opportunity to help Catalyst, a classical example of one of those companies I talked to you before, now a development company looking to grow a pipeline, came to us to reinstitute research. They gave us a protease and they said, can you engineer it to stay in the back of the eye? And so we got together and worked with them on this. So what is geographic atrophy? It's an eye disease that happens in mostly older adults. And what happens is geographic atrophy is really a form of age-related macular degeneration. It's a form of what we call dry age-related macular degeneration. There's a wet form which involves blood vessel growth and edema. The dry form is really just based starts with these drusen that you see here. And the complement particles are part of what drives drusen to form, and eventually it degrades your retina and destroys your vision. And so turns out if you can block complement, you can slow this process from happening. And that's essentially what APL2, which is the Apellis drug, did. And this is just to clarify the complement pathway. This is kind of an old slide because I'm sort of telling an old story intentionally. This was the state of the art at the time. And I'll show you on the next slide, the state of the art is advanced some. But the idea is complement factors C3 there in the middle is sort of the funnel that everything goes through. So if you can block C3, you're really going to shut down the pathway more effectively than hitting many of these other things. Some people think that maybe you want to only shut down one of the three arms of complement, but what Apellis has shown is that C3 is actually a good way to do it when you're hitting them all at the same time. So we were a fast follower to that. Basically, APL2, now a drug called Syfovre, I think is how you pronounce it, is approved for treatment of geographic atrophy. It does indeed slow geographic atrophy. At the time we started this program, it had finished a phase 2 study that looked very promising. But we thought, well, if you had a choice of getting an intravitreal injection once a month, which is what the best therapeutic result you get with APL2 is, wouldn't it be better if you had to come in every 2 or 3 or 4 months? And so we said, well, with a protease, you're going to remove complement many times with one molecule. With APL2 or Syfovre, you bind 1-to-1, right? So if you have a catalytic process, maybe you need less drug around that can last for longer. And so if you just engineer this thing to have 1, a good half-life, and 2, its catalytic ability of cleaving many C3s per molecule, we could have a longer pharmacodynamic effect. And so that was the goal of this program, was to engineer this protease to do that. So as I said, what Catalyst had discovered was a variant. There were multiple mutations in the human protease called matriptase, and they had taken the catalytic domain and changed it from its normal specificity to one that was really very specific for just complement factor C3. And but they hadn't engineered it to stay in the eye. They hadn't figured out how to make it very well. It was very difficult to produce, and you had to refold. And you had to modify it in a way that allowed it to stay in the eye for longer. Now, you probably guys don't necessarily know this, but it turns out bigger things stay in the eye for longer than smaller things do. Guess that's not so surprising. So we wanted to make it bigger. What do? You put a peg on it. We actually thought of many other ways to make it bigger, but it turns out matriptase is a particularly tricky molecule in that as a protein, it does not like if you put anything on either the C or the N terminus. It refuses to function in any of those cases. In fact, it won't even get produced particularly well. So we had to find a way to pegylate it in another place. We did that. As you can see, we get nice, stable monomeric species there on the top. They have equivalent activity in the hemolysis assay. That's a classic complement testing assay. And so we felt like we were now off and running here. So we said, okay, let's put this molecule in vivo. And we started off commonly when you want to see whether you're getting pharmacokinetics that will mimic what happens in humans. Rodent eyes are not so good in that regard. Their just structure is very different than a human eye, but rabbit eyes are a good place to start, and they're much less expensive than non-human primates. So we started with rabbits. And as you can see there, and I see a Regeneron colleague in the audience, they have one of the greatest drugs on the planet. I say that in part because I worked on it when I was there, called Eylea or aflibercept. And that's a classic wet AMD drug, but it's a good pharmacokinetic marker as a control. So we ran 2782 head-to-head, and you can see the pegylated version of 2782 had similar pharmacokinetics in a rabbit to aflibercept. So we were excited about that, and we said, okay, now we can go into monkeys and do not only a PK study again, but a PD study. So it turns out that complement is different enough in other species, not surprisingly, that this protease did not do anything on rabbit complement. And so we did it in non-human primates, and then you can see the pharmacokinetic curve. And then we just asked the question, how long do we suppress C3? How long is it gone for? You can see it's gone for over 30 days. We had actually stopped the study, and we realized that at day 28, we still didn't have any C3 around in the eye. And we called up the CRO and said, you still have those monkeys around? Because this was serial sampling from the monkeys. We want to get another sample from them. They said, yeah, well, actually, they're running around. They're fine. We can get them again. And so we got another sample around day 56, I think it was. And sure enough, we started to see the complement come back. That allowed us to model the pharmacodynamics and predict what happened in monkeys. What does that predict will happen in humans, or from what's known what happens in monkeys? And in fact, we predicted that we would have a much longer half-life than the naked molecule there you see in orange, which would have been similar to the profile that you get for APL2. So we thought we had a really good program here. So this is kind of an end-to-end process. We figured out how to make the protein. We figured out how to pegylate it. We put it in eyes. We measured all the pharmacodynamics, all the pharmacokinetics, and basically provided a full service to Catalyst. Everything they would have done to hire themselves, we did at Mosaic. And it resulted in this molecule being licensed to Biogen. And ultimately, it's now in the hands of Vertex, actually. So it's moved around a little bit. Okay, so I'm going to talk a little bit. How am I doing on time? Can I tell? Oh, okay. Should be able to make it. I'll talk a little bit about our work in antibody discovery and optimization. And I'm going to tell you 5 short stories. One about P95 and how we optimize that molecule to as part of a bispecific. I'm going to tell you a story. So the rest of these stories, unfortunately, are partner programs that we've had. And so I'm going to have to be a little vague about them, but I think I'll give you enough information that you will understand what we did and why we did it. But there'll be affinity optimization that of an antibody binds to 1TM and how we got improve the potency of mouse in parallel with the human increase in potency. One where we had an interesting specificity, dual specificity molecule that we were doing. And because we had done an animal immunization to get the starting antibody, we had to humanize it. And we humanized it and optimized it in the same pot. And there was some interesting, a little story there that came out of that. The 4th one is a discussion around a nanobody. So we have a nanobody library that's an all human, I guess it's an engineered VH domain. It's not really a nanobody. It doesn't come from a camel. It's actually a human framework we've engineered to be like a nanobody and not care that there's no light chain there. We made ones that bound to a glycan on a protein that's produced specifically on a certain cell. And then the last story I'll tell you quickly is about murinization. We talk about humanization. Well, sometimes you want to murinize things, and I'll explain to you why, if that's not already obvious. So here's the P95 story. Essentially, P95 is a truncated version of HER2. You just, in the past talk, a little bit about HER2. It's a really good target for tumors, but it has somewhat of a problem in that, it is on normal cells, and so people are trying to balance dealing with that. But in the case where you have a tumor, those are the only times you really get the truncated version formed. And so that is really truly, as close to a tumor-specific antigen as opposed to a tumor-associated antigen. And so it's a good target to think about for a CD3 bispecific. And, Roche thought of that and published a paper on it that you can see down there. And so we were making this molecule as a tool molecule for us that came out of this paper. And we were getting really frustrated because we were trying to make it and we just could hardly produce any at all. It was just like tearing out our hair, and you can see what's happened now. And what we figured out was, wait a minute, let's look in the patent. And it turns out that Roche was having this problem too with their own molecule. So we said, well, maybe we should actually try out our yeast display and see if we can fix this. And so we set up to do an optimization, and we started to do that. Now, what we used here is error-prone PCR. Now, that's about as old school a way as you can do an optimization as there is. But I like the old school ways, and I particularly liked it in this case. And the reason is that you don't pretend to be too smart when you use yeast display. And that's what I like to do. I like to play dumb at least part of the time because you figure things out that you would have never figured out otherwise. So a lot of people want to just change the CDRs. And, we could have done that, but we wouldn't have got this result. And the reason is that it turns out the mutation that made this antibody produce better was actually in the framework. By the way, this is what nature does too, right? Somatic cell hypermutation. Our bodies do not play any smarter than we have to. They make mutations where you need to make. And actually, there's probably a biological reason for that. If you change the germline sequences in your CDRs to do something else than what they normally do, like bind an antigen, well, that might no longer be the framework that holds those CDRs stably and in a perfect position. And so naturally, evolution in our bodies allows us to make changes elsewhere to compensate for things we might be messing up in the CDRs. So, we're like technology's version of somatic cell hypermutation. In this case, we improved the expression level by 50-fold. We got another 5-fold on the potency. This was just after a couple, 3 rounds of this. And so just as an example, we stopped there. But just to show you that sometimes the old school way is the way to go. Second example is this program where I was telling you about it hits the 1 TM, and we didn't we didn't have really great potency on the mouse, and we wanted to improve it there. So it was cross-reactive. We had 2 parental molecules. In this case, again, we did error-prone PCR, and as you can see, we brought down the potency in both of the optimized molecules, where we now had sufficient murine activity that it could be tested further in those animal models. Sorry, not animal models. First, it was in the biochemical models or cell-based assay models. But you can see the potency was not really good enough in the first molecules, even though it was okay on human, it really wasn't good enough in the mouse. And so just through simple error-prone PCR, we were able to optimize this using our yeast display platform. Okay, third example. So this is the case where we were going after, wanted to bind a molecule that started and had some activity on its own, but could be cleaved into another molecule, and we wanted to block both. So it has somewhat different activities in each case, but we wanted to be able to hit both and block both. Sorry, I'm being a little vague here. And so we did an immunization with Abveris. We like their hyperimmune mouse because we think we can get a broader diversity of responses with that mouse, but it's a fully mouse immune system, so we got to humanize the molecule after that. And so I'm going to tell you a little bit of story where here we tried to be intelligent. And we said, okay, let's try and humanize. Let's make a library where we humanize and make all the changes and tweaks that you might want to make to humanize the molecule, and change the amino acids in the CDRs as well in a design library, so that we could try and optimize the potency as well, because the starting molecule, the parental mouse molecule, was good, but not really good enough. And so as we're going through this cycles of optimization, what we're noticing is the potency is coming down, or the affinity is going up, and the KDs are going down, is what I meant to say. The affinity is going up, but we're finding we're kind of stuck on the humanness of the molecule. It's staying really kind of about the same. It improved a little bit. And we started thinking about this and said, well, we're doing this selection. We're selecting for expression on the yeast surface. We're selecting for binding potency, but are we selecting for humanness? No. And guess what? We're not getting it. It would have been lucky, actually, for us to get it. But what we did know is through deep sequencing, we found out all the amino acids that were giving us the potency increases. And so we knew what those were. And so then we separately humanized the framework by more traditional methods. And it turns out that those sequences that work well in the not very human version actually translated perfectly into the humanized version. And those are the clones you see there on the bottom, where now we've gotten the humanness much further up to where you want to be, and have still way more potent molecules than that mouse parental molecule, or the humanized one itself, where we lost a little bit more than the mouse one, but not too much. Okay, last story. Sorry, next to last story. So this is an example where our partner wanted a nanobody to this glycoform of a cell-specific glycoform of a protein on the surface of these cells. And so we did selections, and you can see we were able to, by round 3 on the figure on the left, get binding to the cells. So this is a cell-based selection, we got binding to the cells, and we had low binding to an off-target protein that was related, but thought we would be a good negative selection. And then from there, we were able to make a purified version of this glycosylated protein. And as you can see, we're able through additional selections to get molecules that bound the correct glycoform that we want, but not the protein itself or the alternate glycoform that happens on different cell forms. So just an example where we can do cell-based selections with this nanobody library and actually bind glycoforms, not even just proteins. Okay, the last example I'll show you before I finish up here is this idea that sometimes you want to murinize things. So often when you do antibody discovery, you guys know this, basically you make a human antibody to the target that you want. And if you're lucky and there's enough homology there, you can get activity against the mouse target as well. The problem is, if you want to test that human antibody in a mouse and the model is going to go for more than a few days or a week, you're going to get in trouble pretty fast. And so what you want to make is the mouse version. So you can make a chimera and leave the human variable domains there, but that's imperfect. So sometimes what you want to do is murinize the molecule. You want to take your human antibody and actually make it into a mouse antibody, even in the variable domains. And so we're able to do that. You can see here, we made a lot of variants. So this is as if we were humanizing. You just make all the same kind of mutations, except you're reverse engineering into a mouse framework instead of a human framework. And you can see we're able to maintain the human potency, even increase it in some cases, and increase the mouse potency as well while we're doing this. So sometimes when you're in the early stages of program and you want to get proof of concept of the biology in a mouse, sometimes it's necessary to do this. And it's just an example of the kinds of things we do at Mosaic. We do what the partner needs us to do to solve the problem of drug discovery, no matter what it is. So that's the end of the story I'm going to tell you. This is just a list of the things. We engineer molecules for long intravitreal half-life. We've done this actually with antibodies as well, pegylated them, and used other techniques. We actually have one story here that I didn't tell. Another partner came to us and said, we want to work in the CRISPR space and we want to change a wild type protein to have a different profile of binding. And what we did, they said, well, we've got to deal with a CRISPR company, but we don't know which mutation to make. So we said, how about if we make you the wild type protein and a bunch of variants, and we'll test all the binding affinities, and we'll find one that has the profile shift that you want. Then you can go back to the CRISPR company that you got to deal with and tell them what guide RNA you want. We did exactly that. We found them exactly which mutations, in this case, a single mutation to make that gave them the profile they wanted, and they went back and now are moving forward. And we've now helped them since then with cell-based assays and intracellular trafficking and pharmacodynamics in animal models. So we're happy to do anything for anybody. If it's an interesting science problem, we like to work on it. We have business models that are mostly fee for service, but we are willing to have a little bit of skin in the game if you want us to do, where there are downstream milestones. We particularly actually like those programs, even though generally people don't like to do it, because the milestone ends up making us more than the discount we give you on the way there. So, and since we don't fail very often, we end up making more money that way. But that would be your decision as a company. So here's the folks we have. Fortunately, these aren't coming out without a little overlap there. I don't know what happened. But the majority of the people have advanced degrees in our company. Just a bunch of scientists that love to do drug discovery with anyone who wants to engage with us. So that's all I had. Hopefully I'm not over time.
We would have time for maybe one or two questions, if anyone has any. And if not, I can start. I was curious about with the C3, so the enzyme to cleave C3, did you explore attaching larger proteins or like larger pegs, or even look into maybe some proteins that would be specific for, say, receptors on eye cells? Or
Yeah, so it's a good question. So in terms of other proteins to attach, you can do that, and we actually do that for other partners who work with us in eye programs. We actually get a fair number of eye programs. The problem with this one is you'd have to chemically attach a protein to the protease, because the C and the N terminus were intolerant to any modification whatsoever. So that's why we really ended up focusing on the pegylation. We did try pegs up to 80,000. The one that we made there was a 40, and we didn't see enough of an advantage going to 80 over 40, and given toxicity concerns from very, very large pegs that there are in the industry. Whether they're real concerns or not is another question, but there's a perceived concern. That's why we stuck with a 40.
Thanks.
Hi, thanks for all the stories. A lot of really fun examples there. So how many programs did you run at a time with your team?
We usually have probably 10-ish that run at any one time. We have 40 people in the company. About half of them are the bigger programs that last years or more. And, maybe we have 4 or 5 or 6 people on them working. And then the other half of them tend to be smaller programs, kind of like the first project that I described we did with the company on the making the mutants and variants of their protein they wanted to do in the CRISPR. We probably have, probably half the programs or that kind of thing, and then they turn over faster.
Do you find you have like a waiting list? Is it? No.
No.
Perfectly. Okay.
Yeah.
Thanks.